Now I would like to introduce Wendy Chen, who is a product manager and will certainly tell you about what can Abcam contribute to in the EMT field.
Fantastic. Thank you, JP. I'm Wendy, and I'm part of the team that's responsible for product management and marketing at Abcam. Many thanks, JP, for such a great presentation and everyone for attending. Whilst you're thinking of questions, I would like to briefly talk to you about our ongoing efforts to drive accurate and reproducible scientific research, and also highlight some of the products that will be of use to you in your EMT research. We know that, as a scientist, you're under pressure. Pressure to publish, pressure to secure funding, pressure to get results. We also know that when experiments fail because of poor quality reagents, it wastes valuable time and resources. That's why reproducibility and reliability are everything to us. We work hard to ensure our reagents give reliable and consistent results. This is most recently seen in our knockout validation initiative to confirm specificity with a true negative control, or in our RAB-map antibodies that offer you the sensitivity you need, and in our recombinant antibodies that provide you consistent results regardless of batch. Providing you with the highest standard of reagents is something we have always believed in, and is central to all our ongoing strategies. Ultimately, we work hard to eliminate the time wasted on repeat experiments so that you can get to where you need to be faster. In terms of products you will be interested in, I would like to highlight some of the validated EMT markers we have. We have all the antibodies you will need to detect the key players of EMT, including E-cadherin, N-cadherin, pan-cytokeratin, fermentin, and all the transcription factors that are so crucial to the EMT process, including twist, snail, slug, and SEP1. Here I have included examples of stains using one of our E-cadherin, N-cadherin, and snail and slug antibodies on various human carcinoma tissues. One antibody I would like to highlight is our fermentin antibody. Fermentin is a type 3 filament protein that is often upregulated in EMT. This rapid monoclonal fermentin antibody is made using a RAB-map technology, which combines the superior antigen recognition of a rabbit antibody with the specificity and consistency of a monoclonal. This particular fermentin antibody is also highly validated in human, mouse, and rat in IHC, IFICC, flow cytometry, and Western blot. It has been cited in over 100 independent publications and rated as excellent by customers in 30 independent product reviews. This fermentin antibody is also available in a BSA and azi-free formulation or conjugated to Lexovir dyes, and a 10 microliter trial size is available for purchase. We've put together a basic EMT panel that is suitable for anyone who's interested in doing a top-level preliminary assessment of EMT. It contains antibodies against the 4 main markers of EMT: E-cadherin, N-cadherin, snail and slug, and fermentin, and 1 goat anti-rabbit secondary antibody. Like JP has highlighted in his talk, there's an urgent need for panels that is suitable for accurate EMT scoring and determine the EMT status in different cell lines, normal tissues, and tumors. We'll be collaborating with JP and drawing on his expertise to establish these panels. If there are any particular cell lines or tumor type you would like us to look into first, please let us know. To keep up to date with Abcam's latest product offerings, I would strongly encourage you to register an account with Abcam. You can do so by clicking on the register link on the top right of the Abcam website. Besides from using antibodies to detect and assess EMT, there's also a need to quantify the proteins involved in the process. Therefore, I would like to introduce our range of SimpleStep ELISA kits. SimpleStep ELISA kits are sandwich ELISA assays that we have developed to be as simple and effective as possible. Unlike traditional sandwich ELISAs, which take more than 3 hours to run, SimpleStep ELISA kits generate data in just 90 minutes without compromising on performance. A conventional sandwich ELISA uses a pair of antibodies that bind to at least two distinct sites on the target protein. The capture antibodies pre-coated onto the well of a microplate and selectively binds to the target protein. After a wash step, the detector antibody is added and binds the second site on the target protein to form a sandwich complex. SimpleStep ELISA kits streamline this process by using homogeneous format, wherein the antibody analyte sandwich complex is formed in solution in one single step. In just one incubation and wash step, the complete sandwich complex forms in the well and is anchored to the plate with an immunoaffinity tag. In addition to drastically reducing assay time, SimpleStep ELISA kits are also one of the most sensitive assays in the market. On the right-hand side here is a standard curve comparison between a human interleukin-8 SimpleStep ELISA kit and a traditional ELISA kit from leading competitor. Interleukin-8 has been shown to induce EMT and maintain the mesenchymal and invasive features of various tumor cells. Our SimpleStep ELISA kit shows a threefold increase in sensitivity than the leading competitor. Some of the other SimpleStep ELISA kits you may be interested in include interleukin-4, interleukin-5, apolipoprotein A4, and PARC7. These kits have all been developed by Abcam as a result of ongoing collaboration with JP and AidStar. For more information on our ELISA kits, please visit www.abcam.com/ELISA. Last but not least, for modulating EMT in different cell lines, we also have a wide range of EMT inducers and inhibitors. For inducing EMT, we have biologically active recombinant proteins of TGF-beta1, EGF, and HFG. We have a great range of products to suppress EMT from our small molecules range as well. For example, all the inhibitors that JP has used are available, including the PI3K inhibitor GDC0941, the SERC inhibitor AZD0530, the Merck inhibitor PD03-25901, the TGF-beta receptor 1 inhibitor A8301, and the c-MET inhibitor JNJ38877605. For more information and resources for your EMT research, please visit www.abcam.com/EMT. With this, I'd like to end my presentation and thank JB again for his great presentation. I will now hand it back to Jessica for the Q&A session.